Angela D. Friederici is a highly acclaimed German linguist, psychologist, and neuroscientist, renowned for her extensive research into how the human brain processes and acquires language. As a founding director and scientific member of the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig, Germany, she has significantly contributed to our understanding of the neural basis of language. Her work often integrates linguistic theory with brain imaging techniques like fMRI, focusing on the roles of areas like Broca's and Wernicke's, and the arcuate fasciculus in speech and grammar processing. Angela D. Friederici was born in Cologne, Germany, in 1952. Her academic journey began in 1970 when she embarked on studies in linguistics at the University of Bonn, Germany, and the University of Lausanne, Switzerland, earning her PhD in linguistics in 1976. Parallel to this, from 1975 to 1980, she also pursued psychology at the University of Bonn, completing a diploma in psychology in 1980. She further solidified her academic foundation by obtaining her habilitation professorial dissertation in psychology from Justus Liebig University Gießen in 1986. Her early career included a postdoctoral scholarship at the Massachusetts Institute of Technology (MIT) and research work at the Aphasia Research Center in Boston. She then spent a significant period from 1979 to 1989 as a research fellow at the Max Planck Institute for Psycholinguistics in Nijmegen, Netherlands, and also conducted research at the Université René Descartes in Paris and the University of California, San Diego. In 1989, she became a full professor for cognitive psychology at the Free University of Berlin before becoming a founding director and scientific member of the Max Planck Institute of Cognitive Neuroscience, now the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig, in 1994, where she has continued her groundbreaking work. Friederici, A. D. Pfeiffer, E. and Hanna, A. 1993. Event-related potentials during natural speech processing. Cognitive Brain Research, 183 to 192. This is often cited as one of the very first papers to report the ALA, specifically in response to phrase structure violations in natural speech, laying the groundwork for subsequent research on its role in early syntactic processing. Friederici, A.D. 1995. The Time Course of Syntactic Activation During Language Processing, a model based on neuropsychological and neurophysiological data. Brain and Language, 53, 259 to 281. In this theoretical paper, Friederici proposes a multi-stage model of language comprehension, where the ALA is explicitly posited as reflecting the initial, automatic phrase structure building process. This paper helped solidify the ALA's theoretical significance. Hanna, A. and Friederici, A.D. 1999. Electrophysiological Evidence for Two Steps in Syntactic Analysis, Early Automatic and Late Controlled Processes. Journal of Cognitive Neuroscience, 193 to 204. This study further elaborates on the dual-stage model of syntactic processing, with the ALA representing the early, automatic detection of word category violations, and a later P600 component reflecting more controlled reanalysis. Steinhauer, K. Alter, K. and Friederici, A.D. 1999. Brain potentials indicate immediate use of prosodic cues in natural speech processing. Nature Neuroscience. 191 to 196. This foundational paper investigated how the brain processes prosodic cues, such as intonation and rhythm, in real-time speech comprehension. Using event-related brain potentials (ERPs), the authors demonstrated that prosodic information is not just an ancillary feature but is immediately and actively used by the brain to guide the processing of spoken language, influencing syntactic parsing from very early stages. This challenged previous views that might have emphasized a more sequential or delayed role for prosody. Mies, B. Kolsch, S. Gunter, T.C. and Friederici AD. 2001. Musical syntax is processed in Broca's area, and Meg study. Nature Neuroscience. 4.5, 540-545. This study provided groundbreaking evidence for a shared neural substrate between language and music processing, specifically focusing on syntactic aspects. Using magnetoencephalography MEG, the researchers found that violations of musical syntax elicited activity in Broca's area, a region classically associated with language processing and grammatical structure. This suggested that the brain's capacity for processing complex structural regularities might be more general than previously thought, extending beyond linguistic domains to other structured systems like music. Friederici, AD. 2002. Towards a neural basis of auditory sentence processing. Trends in Cognitive Sciences. 6.2, 78-84. In this review article, Angela Friederici synthesized the burgeoning research on the neural mechanisms underlying auditory sentence processing, proposing a comprehensive model of how the brain handles spoken language. The paper laid out a neurocognitive architecture that delineates distinct processing stages, including initial phrasal structure building, thematic role assignment, and integration of semantic and syntactic information, and mapped these processes onto specific brain regions, particularly within the left hemisphere. Friederici, A.D. Steinhauer, K. and Pfeiffer, E. 2002. Brain Signatures of Artificial Language Processing, Evidence Challenging the Critical Period Hypothesis. PNAS. 99-1, 529-534. This study challenged the strict interpretation of the critical period hypothesis for language acquisition by examining how adults learned an artificial language with complex grammatical rules. The researchers observed that even adult learners exhibited brain responses ERPs indicative of rapid and implicit processing of the artificial grammar, mirroring those seen in natural language acquisition, particularly for syntactic violations. Friederici, A.D. and Kotz, S.A. 2003. The Brain Basis of Syntactic Processes, Functional Imaging and Lesion Studies. Neuroimage, 20 Sup 1, S8 S17. This review paper discusses the neural underpinnings of syntactic processing, where the ALA is a central electrophysiological marker associated with early structure building processes, linking it to findings from fMRI and lesion studies. Opitz, B. and Friederici, A.D. 2004. Brain Correlates of Language Learning, the Neuronal Dissociation of Rule-Based versus Similarity-Based Learning. The Journal of Neuroscience. 2439, 8436-8440. This research explored the distinct neural mechanisms involved in different types of language learning, rule-based learning explicitly learning grammatical rules versus similarity-based learning implicitly learning patterns from exposure. Using fMRI, the study demonstrated that these two learning strategies recruit distinct brain networks. Rule-based learning engaged prefrontal and parietal regions associated with explicit cognitive control and working memory, while similarity-based learning activated more implicit, procedural learning systems, highlighting the brain's flexible approach to acquiring linguistic knowledge. Friederici, A.D. 2004. Event-Related Brain Potential Studies on Language Processing. Handbook of Psychophysiology, 236 to 261. As part of a handbook, this chapter provides a comprehensive overview of ERP research in language, with a significant focus on the ELA as a key component reflecting initial syntactic parsing. Friederici, A.D. Ballman, J. Heim, S. Skubitz, R.I. and N. Wander, A. 2006. The brain differentiates human and non-human grammars, functional localization and structural connectivity. PNAS 103-7, 2458-2463. This influential paper investigated the neural processing of human-specific grammatical rules compared to simpler, non-human grammars. Through fMRI and diffusion tensor imaging DTI, the researchers found that while both types of grammars activated a common set of brain regions, human-specific recursive grammar uniquely engaged a specific part of Broca's area pars opercularis and its connections to the temporal lobe via the arcuate fasciculus. Friederici, A.D. Fiebich, C.J. Schlesowski, M. Bornkessel, I. and von Kramen, D.Y. 2006. Processing linguistic complexity and grammatically in the left frontal cortex. Cerebral cortex. 1612, 1709-1717. This study further delineated the roles of different subregions within the left frontal cortex in processing linguistic complexity and grammatically. Using fMRI, the authors showed that the posterior part of Broca's area Brodmann area 44-45 was primarily involved in processing syntactic complexity, while the more anterior frontal regions e.g. BA47 were engaged when grammatical violations occurred, particularly those affecting semantic integration. Friederici, A.D. von Kramen, D.Y. and Kotz, S.A. 2007. Role of the corpus callosum in speech comprehension, interfacing syntax and prosody. Neuron. 53-1, 135-145. This paper highlighted the critical role of the corpus callosum, the large white matter tract connecting the two cerebral hemispheres, in integrating different aspects of speech comprehension. Specifically, the study demonstrated that the anterior part of the corpus callosum is crucial for coordinating syntactic and prosodic information during language processing. Damage to this region impaired the ability to use prosodic cues to resolve syntactic ambiguities, underscoring its importance in the seamless integration of these distinct linguistic features. Makuchi, M. Ballman, J. N. Wander, A. and Friederici, A.D. 2009. Segregating the core computational faculty of human language from working memory. PNAS 106-20, 8362-8367. This research aimed to distinguish the neural networks underlying the core computational aspects of human language from general working memory processes. By manipulating linguistic complexity and working memory load independently, the study found that a specific part of Broca's area Brodmann area 44 was selectively activated by linguistic hierarchical complexity, even when working memory demands were low. In contrast, other frontal regions were more involved in general working memory. Friederici, A.D. Kotz, S.A. Scott, S.K. and Abelzer, J. 2010. Disentangling Syntax and Intelligibility in Auditory Language Comprehension. Human Brain Mapping. 31-3, 448-457. This study investigated how the brain processes syntactic structure independently of speech intelligibility, a common challenge in auditory language research. By using degraded speech that maintained syntactic structure but varied in intelligibility, the researchers were able to dissociate the neural responses to these two factors. They found that while intelligibility primarily engaged bilateral superior temporal regions, syntactic processing, particularly the detection of grammatical violations, consistently activated left inferior frontal regions Broca's area, further solidifying its role in processing grammatical structure, even under challenging listening conditions. Friederici, A.D. 2011. The Brain Basis of Language Processing, From Structure to Function. Physiological Reviews, 91-4, 1357-1392. While a broad review, this paper reiterates the foundational role of the ALA in the initial phase of auditory sentence processing within her neurocognitive model of language. Since 2011, Angela D. Friederici has continued to be a highly prolific and influential researcher in the cognitive neuroscience of language, extending her foundational work on the neural underpinnings of language processing. Her publications in this period often reflect a deepening focus on the neurobiological basis of human-specific language faculties, particularly syntax, as well as its development and evolution. For instance, a seminal review in Physiological Reviews 2011 consolidated her model of language processing, outlining the frontotemporal networks supporting syntactic and semantic processes, and later extended to explore the evolution of the neural language network in Psychonomic Bulletin and Review 2017. Her 2017 book, Language in Our Brain, The Origins of a Uniquely Human Capacity, synthesized many of these later insights, integrating adult language processing, development, and brain evolution. Her work has also delved into the maturation of the language network in children, as seen in PLOS1 2011 and Cerebral Cortex 2019 studies on cortical surface area expansion and white matter myelination. Furthermore, she has continued to investigate the neural mechanisms underlying fundamental linguistic operations like merge, a concept from Chomskyan linguistics, examining how specific brain regions, particularly within Broca's area and its connections, are dedicated to processing hierarchical linguistic structures, as highlighted in papers like her 2023 article in Trends in Neurosciences on the evolutionary neuroanatomical expansion of Broca's region. References: Professor Angela Friederici, The Neurobiology of the Human Language System. July 25, 2018. Retrieved February 4, 2019. Angela D. Friederici CV. Max Planck Institute for Human Cognitive and Brain Science. Retrieved February 4, 2019. Friederici, A. D. Pfeiffer, E. Hanna, A. 1993. Event-related potentials during natural speech processing. Cognitive brain research. 1-3, 183-192. doi 10.10160926-641093900026-2. PMID 825-7874. Friederici AD. 2004. Event-related brain potential studies in language. Current Neurology and Neuroscience Reports. 46, 466-70. doi 10.1007S11910-004-00700. PMID 15509448. S2 SID 46656614. Friederici, A Language in Our Brain. MIT Press. 2017 Leibniz Prize. Retrieved February 4, 2019. Angela Friederici ist in Habrinder 11. Johannes Gutenberg Stiftungsprofessor Angela Friederici is the winner of the 11th Johannes Gutenberg Endowed Professorship in German. November 10, 2009. Retrieved February 4, 2019. Award of the Wilhelm Wundt Medal of the German Psychological Association DGPs 2018 to Professor Angela D. Friederici. September 24, 2018. Retrieved February 4, 2019. LSA elects 3 to honorary membership. Linguistic Society of America. January 24, 2019. Retrieved February 4, 2019.